So I'm going to introduce Dr. Antoni Ribas. He's a professor of medicine, surgery, and molecular and medical pharmacology, and director of the Cancer Center Tumor Immunology Program area. He's been here with us since 1998, has published over 200 scientific peer-reviewed manuscripts and clinical and laboratory research in malignant melanoma. And you guys may know his name from all the New England Journal papers that he's been published along this last few years. So please join me to welcome.
Thanks, Satiro, and it's great to be talking here. So here's the outline of what I want to talk about. So we're going to start with a checkpoint inhibitor immunotherapy. There were some references from in the prior talks, and then we'll do one part of this area that we're interested, which is the combination of immunotherapy and targeted therapy, and then we'll talk something that's more related to the theme of this symposium, which is T-cell receptor engineering. This is the whole concept, which is many cancers auto-vaccinate. There's some antigens that are immunogenic that lead to an immune response, but our immune system has limited those with a series of checkpoints or inhibitors or breaks to the immune system. And these are spatially regulated. Whenever a T cell needs to be licensed to kill, it requires two signals: immunological signal 1, which is the antigen, and immunological signal 2, which is co-stimulation. And that happens when a dendritic cell is trying to train a T cell. But to self-antigens, that's usually on the off switch where the T cells are turned off by CTLA-4, which displaces co-stimulation. So when this was recognized by Jim Allison and Jeb Bluestone and others, then it led to the development of antibodies that block this breakup here. And then the T cell gets the two signals, then it can start dividing, circulate through the blood, and then target whatever it's supposed to be targeting. This is not a tumor antigen specific approach. This is very broad. So the issue is that this will lead to responses, but also it's likely to lead to side effects. We know this is true because we've incorporated molecular imaging to be able to get the spatial resolution of what happens in a patient that receives a CTLA-4 blocking antibody like tremelimumab. This is because we work with Johannes Cernin, Martin Auerbach, Caius Radu, and others, and Anna Wu's here, who's done studies, and Richard Tavare that I'm not going to talk about, but go to the same kind of idea that we can use molecular imaging to study population dynamics. And this is in patients that we treated some years ago, where we have two patients at baseline and the same two patients with a scan at around a month later. And we did this for around 10 patients. And we use a PET tracer that's not your regular PET tracer. This is FLT or thymidine. And thymidine is what any of you are in the lab are using to see if cells divide in vitro. You add radioactive thymidine and you see how it's incorporated into the DNA or RNA. So we can do the same in humans. And we can see that the most uptake is in the bone marrow because that's where most dividing cells are. In the spleen, which is the largest lymphoid organ, there's very little uptake. But if we give the CTLA-4 blocking antibody, then we can see that there's an increase in the pink color here and the warm colors compared to here. And that's because cells start proliferating, as the concept would suggest. And that happens in around 2/3 of the patients. If the cells proliferate, then they have to make it to blood, and then they have to make it to where they go. And to look for them in blood, we used an indirect technique, which T-cell receptor sequencing, where we could count the number of T-cell receptor clonotypes that were circulating in blood. We did this in the lab with Liau Robert, and then we collaborated with Larry Fong, and the results are more or less the same, which is the black bars are the healthy donors. All of these is that it denotes that in around half of the patients who get CTLA-4 blocking antibodies, there's a diversification of the number of T cells that are in blood because we can count them by their T cell receptor. And if we look in this different plotting on patients with prostate cancer, melanoma, you can see, going up is the ones that diversify, going down is the ones that decrease the diversification. You can see there's a lot more going up than going down, showing the same kind of idea that in a large subset of patients, if you block CTLA-4, you get proliferation and diversification of the T cells in blood. The more important thing is, do they make it into tumors? So here's studies that we did with Alistair Cochran and Rongrong Huang some years ago, where we have baseline biopsies of patients who responded or patients who did not respond. The little brown dots are CD8 positive T cells, the effector arm of a cellular immune response. And at baseline, we couldn't see much difference between responders or non-responders. When we did biopsies a month after getting CTLA-4 blockade, we could see that the responders had a great increase of T cells in the tumor, which makes sense. Those are the effectors. If they don't increase, then there's not going to be an anti-tumor response. But patients who did not respond, there was also an increase of T cells. And we can see that when we quantified the cell density, we'd see that in 2/3 of the cases, there was an increase from the baseline biopsy to the post-dosing. The two open symbols are the patients with an objective response, telling us that it is very frequent to have T cells turn on the right way and made it into the tumor. But there's only a 10% response rate with this therapy. And here are the randomized trials that led to the approval of ipilimumab in melanoma. You can see that the patients on ipilimumab do better than the controls, but it's just the percentage, is around 10%. So even though we get evidence of mechanistic effects in 2/3 of the patients, and we get T cells into tumors, only 10% of the patients respond. And now we know why that is. It's because when the T cell gets into the tumor, it makes interference, the tumor protects itself through the expression of PD-L1, and there it tells us that we have to do something else. We have to block this late checkpoint on when the T cell is trying to attack the tumor, and here would be then the T cell is turned on because we block this negative regulator. So when we do this, then we have even better clinical outcomes. These are studies with one of these anti-PD-1 antibodies, Nivolumab in melanoma, compared with the carbazine with chemotherapy. Nivolumab does much better than the carbazine. This is another one of these antibodies, pembrolizumab, compared to ipilimumab. Ipilimumab is this arm, was already better than the carbazine, so it's already starting higher. And we can see that PD-1 blockade gives a better outcome, and these are overall survival curves. So even though many of the patients in the control arm ended up receiving PD-1 blocking antibodies, the survival was always better by starting with a PD-1 blocking antibody. I can show you a lot of cases, a lot of things about this, but this is just one case, and I'm showing to you not only because of the scans with this large mass that responded and this large liver mass that was taking over most of the liver that responds with just blocking this PD-1 on a patient who had progressed on every possible therapy at that time. That was in 2012. I just saw him this last week, so 3.5 years later continues to respond. But this is how this patient started. This was a patient who had progressed on biochemotherapy, high-dose IL-2, ipilimumab. There were no other options. The tumor was eating up on his body. You can see that his muscles are nearly gone. He has muscle wasting. He fell and hit his head. I didn't want to do an MRI of the brain because if I found a brain metastasis, that would exclude him from the clinical trial. So I did not do an MRI of the brain. And here's the patient afterwards. We just blocked one thing in his T cells that were limiting those T cells to attack the tumor. And now we can do pamphlets and promotional pamphlets with him. We know why this works. The T cells were there. They had the right receptors. They had escaped all of the control mechanisms that they needed. They got into the tumor. They make interference, and so far all of this sounds good. But then the tumor finds a way to protect itself by taking up from the interferon receptor and expressing the ligand to PD-1, which turns off the T cell. And if we find this, we would assume that those are the patients that will respond. So with Paul Toomey and others, we started testing this hypothesis in patient-derived biopsies. And we did it by staining for the tumor with S100, staining for CD8 cells, which are these cells with CD8. We started recognizing that there was that the patients who responded had a particular distribution of these CD8 cells in something that Jerome Gallon and others had called the invasive margin, which is in between the stroma and the tumor. The T cells do get a little bit into where the tumor is. So if these are sequential cuts, you could see that the tumor goes into here. So they're trying to do what they're supposed to be doing. And if we did serial biopsies on patients, we had 22 patients on the study. No, I'm sorry, 46 patients, 22 with response and 24 without a response. We could see that at baseline, this is the invasive margin area. This would be the tumor. We see T cells outside of this invasive margin. When we release PD-1, they start invading. It goes up in the biopsies at different time points. Some patients, it's low and stays low for a while, and then in a delayed fashion, it goes up. But the patients who do not respond did not have T cells and did not go up, besides one outlier. That's because whenever we block those T cells that were there, that were pre-existing, start dividing and infiltrate the tumor. And we have images about those T cells going from a small lymphocyte to a lymphocyte that's 3 times larger with a large nucleus with Ki-67 uptake. And when we started recognizing these, then we can start thinking, can we use this to predict what happens to patients? Can we just look at the biopsy and say, well, this patient is likely or unlikely to respond? And if you look at this biopsy that has a whole bunch of CD8s, the CD8s express PD-1 because it's an indirect measure of their T cell receptor being triggered. All of that area is full of PD-L1. That's an indirect measure of making interferon. And if we detect this, I would assume that this is a patient who responds. And if we detect this other thing, which is there's no CD8s, no PD-1, and I don't care if there's PD-L1. This PD-L1 is not secondary to an immune attack. I think this patient will not respond. And actually, that's what happens. The patient with this biopsy responded. The patient with this biopsy did not respond. So we think are we that intelligent, and can we test blinded? And we try to do that. We asked Caroline Robert, a dermatologist in Paris, to send us 6 unstained slides from 15 cases of patients she had treated, just the baseline slides, tell us nothing about the patients. We'll look at the baseline biopsies. We'll stain for CD8 and tumor, and then we'll quantitate the CD8s in the invasive margin, and that gives a number, a low number to a high number. The low number means low density, high number means high density. And then we looked at what that would correspond with our set of initial biopsies, and then we would say, well, if there's a low probability of response, then those are patients who are probably progressing. High probability of response patients are probably responding. And then one day we sent an email to each other. We unblinded the series. We told Caroline we thought these patients progress, and she said, well, yes, right. The patients with low CD8 density progress. These patients who had a lot of CD8s, we thought that they would respond. Those patients responded. And actually, Paul only did not, could not predict one of the cases, which thought there was a response and had progression. So that's very good. 14 out of 15 could be predicted just by looking at the baseline biopsy and doing very simple stains, which tells me that we're going into an era where it's not going to be, is the tumor BRAF mutated or not, or GFRB3 or HER2/neu, or I don't know what else. Is the tumor interacting with the immune system in a way that we have to just unleash it? And if that's the case, it would be this one, and most likely it's going to be PD-1, so you just give PD-1 single agent. And then is instead the tumor not inflamed, not have T cells in there? It's maybe not immunogenic, or the T cells were stopped somewhere else with another checkpoint, or did not exist. And then we want to either bring them into tumors, and combining with CTLA-4 would be great. You saw the studies before. We can get a lot of T cells into tumors. Combine with immune-activating antibodies, cytokines, inject things into the tumor. And I see when Julia Scoven is here, she's developing studies where we're injecting into the tumor and then be able to then unleash them. block macrophages or combine with targeted therapy, something that CWEN has also been doing, I'll show in a moment. And in the cases where none of this is going to work because they don't have the right receptors, what Lawrence Cooper and others have pioneered, which is then you just create those cells, make an army of those cells by genetic engineering. So in melanoma, we've got all of this development of immunotherapies, but also we've understand the biology. We know that 50% of melanomas are driven by BRAF, and we can block that with just pill therapies and get very high response rates, but those responses last for a period of time until the tumors progress. So the idea is, can we combine it with immunotherapy, which has lower response rates, but tend to be durable because the immune system, when turned on, has memory and leads to long-term responses. So CWEN by Blanca did these studies where they compare the effects of a BRAF inhibitor, the dabrafenib, a MEK inhibitor, trametinib, with PD-1 in a tumor that's relatively resistant to all these three drugs. But when they put two drugs together, you get better responses. When you put the three drugs together, you get much better responses. And then a series of studies showing that this is reproducible and the mechanism of how this works. And that has allowed us to convince industry that it makes sense to do triple therapy combinations. And the first study we reported at ASCO this year, where we used the dabrafenib, trametinib, the same drugs that CWEN used, with the PD-L1 antibody in a group of patients with BRAF mutant melanoma. And this is what we call a spider plot, where the tumor size starts here. If they progress, they would go up. And if they respond, they go down. So all of these patients go down or stay stable. There was not a single patient who progressed up front to this triple therapy. All of them had either stable disease or an objective response. And the key thing is going to be, are we achieving more durable responses, something that we still don't know. We have to follow these patients much longer to know, are we achieving something that the BRAF and MEK inhibitor by itself will not be able to achieve. But then there's going to be the times where there's no T cells around, so we have to create them. And that's where the concept of T cell receptor engineering comes from. And it's going to be pretty soon 10 years that David Baltimore and Owen Weedy and Jimmy Conomo called us to Caltech and said, well, we would like to set up a program of genetic engineering of T cells and giving them to patients. And we've been meeting frequently with Satiro. Where's Satiro? Somewhere around here. On the right? Okay. Okay, so oh yeah, there's Satiro with a big smile, and Don Cohn and others have been heavily involved in this program of taking concepts from the lab to the clinic and being able to do clinical trials, which is what I'm going to talk about. And the idea came from not only doing genetic modification of the mature T cells, but programming the hematopoiesis to have T cells that are genetically engineered. And that's something that Lily Young, who's now a faculty member here, and David Baltimore had worked on some years ago. But the first approach was we had to start with the simple one of getting a virus to infect the T cells and then give them the receptors and then be able to know what they do. And this is something that first done by Tinley Chodon and Begonia Comín, and now with Beata Bren-Maos, of taking patients' leukapheresis, and in just a very short period of time of 6 days, we expand those cells, we infect them with the retrovirus, give them the T cell receptor, we blood release test them and give them back. We do this while the patient is getting conditioning chemotherapy, and we get 1 billion cells back to the patient. And here's the approach. We genetically modify, put them in a bag. They get admitted in this hospital. We'll condition them with Cytoxan and fludarabine, and then we give the cells back. And we know this is therapeutic. We've published on 15 cases, and here's a PET CT at baseline. This is melanoma in the heart muscle. This is when it goes through the ventricles, but this is a mass in the heart. Really bad. This is a retroperitoneal mass, an adrenal mass, and you can see that just 35 days later of giving 1 billion of these T cells redirected to MART1, we can get response in the heart and in these retroperitoneal masses. When we do it with MARC1 TCR, we learn what Steve Rosenberg had learned, which is there's quite a bit of toxicities of cytokine storm and systemic rash because it attacks also the normal melanocytes, and these patients lose all of their melanocytes and lose all of their pigment. But in the way we had several patients that were really sick. So we turned into what we thought was a safer TCR, MISO TCR, which is expressed highly in synovial sarcoma and a little bit less in other cancers. And with Arun Singh and Bartosz Chmielowski, we've been enrolling on this study. Here's the first 4 patients with the I haven't updated this, but these 2 patients continue to have durable responses going on for a year and a half at least. Here's the first patient with lung metastases. Started responding, and now she is in a complete response. That was in June of 2014, October 2015, she continues to have a complete response. There's another patient with a synovial sarcoma who was progressing on prior therapy with a response and continues to respond. But this is not good enough. We want to do more. And one of the ideas is that maybe those T cells that are giving to the patient are losing function, and we know that this is the case because with Jim Heath and Chao Ma and Wei Wei, who's in the audience, we had been studying the functional characteristics of the T cells are given to patients and what happens when we recover them. Once we recover them, they have lower functionality. So we thought that maybe we could improve that by giving Ipilimumab, the CTLA-4 blocking antibody. So we started a clinical trial with that, and this is the PET trace in the ventricle. This is the mass in the mediastinum. You can see here a partial response. This was a patient with melanoma who had a patchy expression of NY-ESO. The patient responded for a period of time and then progressed. But we know that we can make these cells, we can expand them at the peak. 50 to 60% of the T cells in peripheral blood are transgenic T cells. This is MARC1 tetramer, this is the green. Both CD4s and CD8s are mostly the green cells. With time, we have more black dots as the bone marrow recovers. It starts making more normal cells and decreasing the transgenic ones, so they go down, but they can persist long term with this very short period of manufacture. But what I was talking about before is studies that Jim Heath and Chao Ma did, which is studying the functional characteristics of these cells by applying nanotechnology and microfluidic devices. And I'm not going to show you the data, it's all published, but the bottom line is that the T cells that we give to patients, the transgenic T cells that we give to patients, lose function over time. And this is this would be the green cell. The green cells are the good ones, and then they start losing function. They become purple. They go from actual Th1 to Th2 phenotype. So the hypothesis then goes back to what do we do about this? And the thought was, what if we at the same time genetically modified the stem cells and would allow to have a progeny of T cells that come from the genetically programmed stem cells, and that would allow them to persist long term and be functional. And as I said, this is work by Lily Young and David Baltimore that they published already. Jerry Zak and Dimitrios Batakis have done studies in humanized mice. And then I'm showing you work by Don Cohn using another type of humanized mice and showing that whenever you put the transgenic T cell receptors into stem cells, they become the T cells that come out from that exclude the rearrangement of all the TCRs, so you have like the human, it would be a human counterpart of the TCR transgenic mice, where you can make a whole bunch of T cells that are fully functional, that are with kappa T cell receptor. So we've been working on this, and so the idea that then we put the T cell receptor here, supposed to give it to the mature cell, and then with time, the T cell receptor will be expressed in all of the lineages of hematopoiesis, but it will only be expressed on the cell surface and be functional on the CD8 cells, because the T cell receptor requires a complex, which is the CD3 complex, which is only on T cells. So the rest of the hematopoietic cells will just have the T cell receptor chains intracellularly, and then they'll just probably destroy them. And we have a big group funded by a CIRM grant, a California Institute of Regenerative Medicine grant. We meet every Tuesday, and this is the great group of people who are advancing this project and taking it from the lab into the clinic. And the biggest credit would go to Paula, Beata, to Chris, to Julia, to Angel, and to Begonia, and all of the people who are allowing us to be able to do this. So with this, I want to then give credit to our funders, CIRM, the National Cancer Institute, that funded this, the nanotechnology grants. Owen Whitty and David Baltimore have been very supportive of these projects, and every time we needed a boost of funds, they found a way to do that. Stand Up to Cancer is also funding us for this adoptive cell transfer therapy. We've done this with Jimmy Conomo, Owen Whitty, Don, Jerry, Lily, Arun, and Bartos are working very hard on the clinical studies, and then Jim Keith and David Baltimore, many others involved, and all of the people in my life that I've been citing. So I'll be happy to take any questions. Please, right?
How diverse are the antigens that are really targeted immunotherapy? I mean, are they, I guess I'm trying to think about why carcinomas don't respond to immunotherapy as well as melanomas. And is it due to the antigenic repertoire?
For the ones who haven't heard, the question is why do epithelial cancers not respond as well as melanomas to these immunotherapies? One of the reasons may be just trivial, which is there were a lot more therapies for many cancers than for melanoma, so we were able to incorporate these therapies much earlier in the course of the disease. But we're seeing for checkpoint blockade therapy that the highest activity is where it should be, which is on carcinogen-induced cancers that induce a lot of mutations. And those mutations have the chances of being immunogenic, and then the T cells can better differentiate the cancer cell from the normal cell. It also works in viral-induced cancers, liver cancer, Merkel cell carcinoma. There are very high response rates in those cancers with PD-1 checkpoint blockade. In some cancers, you may need the combination of turning on the immune system and releasing these breaks. I'm sure we're going to be figuring out a lot of these, but you work in lung cancer. These drugs are now approved in lung cancer, and they work on smoking-related lung cancer as opposed to the EGFR mutant lung cancer, which has a lot lower mutational rate. I don't know why it doesn't work as well in breast and prostate cancer, but probably it's a lower mutational load.
So it's the mutational load that's the convergent paradigm?
In general terms, yes, but it's not an absolute term. So there's low mutated tumors that respond. The best example is kidney cancer, which has a low mutational load, but does respond to PD-1 and CTLA-4 blockade. So we'll have to figure out what are the other variables. But if we think of the different variables, one of the important ones will be how immunogenic is the tumor, with either higher mutations or viral infections, viral antigens. Warren?
Good story as well. I appreciate it. So I love these images of the T cells sort of poised on the edge of the tumor, and then there's a sort of, with this release of inhibition, there's a sense those cells can get into the tumor. So when I see those pictures, then I start to think about like what's the immune response in the context of an abscopal response? So in other words, when you are, let's say, treating like Ron Levy's doing at a local level, and potentially delivering local regional release of checkpoint, then you have a systemic effect. And is it your hypothesis in that situation then that you just have to break tolerance or release the immune response at the local level and those can traffic across the body? Or do you need sort of global checkpoint blockade where all lesions have to be bathed in anti-PD or anti-PD-L1, et cetera?
Honestly, I'm quite puzzled by ipilimumab or CTLA-4 blocking antibody working locally by injecting into tumors. Ron Levy from Stanford has reported this in follicular lymphomas, which probably is a low bar to get an immune response. But he tells me that it works in other cancers. So that would probably say that in the cartoons that I generated, there may be something wrong because you can block CTLA-4 in the tumor and then also get the effect. T cells circulate, so maybe you inject locally and then they go to a lymph node and then that's where they get activated. But in our PET imaging, I think it's from human studies would be the one that convinces me the most that the schematic is probably right. We looked at tumors very carefully to try to see, do we see any change of FLT uptake? Yeah. And we couldn't. We also did FDG the day before or the day after. We couldn't see any change, suggesting to me that the replication of T cells with CTLA-4 blockade is in the lymphoid organs. But for PD-1, it's very different. We stain in the tumor, we look for Ki67, we see a remarkable increase of Ki67 positive cells that are replicating in the tumor. So I think in general terms, the schematic has to be right. Why would it work by local injection of ipilimumab will be something that will need to be figured out and studied maybe in these ways. Thank you very much.